Hey everyone, this is Narendra and you're watching to my channel BioGuru. Alright, so now in this video we are going to study about the pigments involved in the photosynthesis. 4 types of pigments may be present in the leaves. How many types? 4 types. What are those? Chlorophyll A, chlorophyll B, xanthophylls and carotenoids. Here the chlorophyll A, its molecular formula is C55H72O5 N4Mg and it imparts blue-green color. Alright, so the next one, chlorophyll B, its molecular formula C55H70O6 N4Mg and it imparts yellow-green color. Alright, next one, xanthophylls. Next type of pigment is xanthophylls. Its molecular formula is C40H56O2 and it imparts which color? Yes, it imparts yellow color. Whereas the carotenoid, its formula is 6C40H56 and it imparts which color? Yellow orange color. Exactly. So how many types of pigments may be present in the leaves? 4 types. What are those? Chlorophyll A, chlorophyll B, xanthophylls, then carotenoids. Alright, now there are different types of pigments present that we understood. Now what are the role of these different pigments? Basically, when pigments are present, their role is to absorb light energy. Now the different pigments absorb light energy at a different wavelength, right? So now that we are going to study under the heading that is called as absorption spectrum. Alright, so here what is absorption spectrum? Absorption spectrum is the graph plotted against the fraction of light absorbed by the pigment. What is absorption spectrum? It is the graph. What it is? It is the graph plotted against the fraction of light absorbed by the pigment. Here it is the graph. This is x-axis. Along the x-axis, we have taken the wavelength of light and written in the form of nanometer. We know that the visible light falls in the range of 400 to 700 nanometer. It falls in which range? 400 to 700 nanometer. Right? And that color, what is that color? Can you tell me? Those colors, short form, they are known as VIBGYOR, right? That we have seen in our previous video. Violet, indigo, blue, green, yellow, orange and red. That falls in this particular range of wavelength, right? And along the y-axis you can see the absorption of light by chlorophyll pigment, and other pigments also. So basically what this graph is showing, it is showing the absorption of light at a different wavelength. Absorption of light at different wavelength by the pigments. Here different types of pigments are there. What is the role of these pigments? To absorb the light. So different pigments absorb light at a different wavelength. So if you put a graph where you will see different peaks at a different wavelength. So this peak is showing the absorption of wavelength or particular color by the pigments. So this graph is called as what? Absorption spectrum. What is absorption spectrum? It is the graph plotted against the fraction of light absorbed by the pigment. Fraction of light absorbed by the pigments. Here if you observe this particular peak, this particular peak is of chlorophyll A. This peak of which pigment? Of chlorophyll A. If you look at the peak, here the peak is lying in the range of 400 to 500 nanometer, particularly near to 400 nanometer, right? Then this chlorophyll A molecule is showing maximum absorption at the 400 nanometer. Then afterwards it is going to decrease its absorption towards 500 to 600 nanometer, and then once again if you see, it shows maximum absorption towards 700 nanometer. That means 400 to 500 nanometer particularly comes between violet and green color, whereas 700 it is towards red color. That means chlorophyll A molecule is going to absorb large amount of violet, indigo and blue color as well as red color. Understood? So what this peak is indicating? This peak is indicating the absorption of particular wavelength of light. So at which wavelength it is coming? Highest peak at around 400 nanometer. So this peak is of what? Which pigment? Chlorophyll A. Not only towards or near the 400 nanometer, but also it is showing a peak towards the 700 nanometer. Clear? So there is one more peak over here. So this peak is shown by which pigment? Which pigment it is? Chlorophyll B. Which pigment? Chlorophyll B. So chlorophyll B, somewhere it is showing the peak. That means it is having maximum absorption nearby 500 nanometer. And then it decreases its absorption and it shows once again another peak. That means it also absorbs the wavelength near to 600 nanometer. Clear? So there is a one more pigment that is of carotenoid. It shows peaks somewhere around 500 nanometer, and then it is not going to absorb in any other wavelength. So what is this graph? This is of absorption spectrum. Now what is absorption spectrum? Once again, it is the graph plotted against the fraction of light absorbed by the pigment. Fraction of light absorbed by the pigments. Which graph it is? Absorption spectrum. What is absorption spectrum? Graph plotted against the fraction of light absorbed by the pigment. Clear? Okay. Next, let us have a look at the one more type of spectrum. Here, action spectrum. Which spectrum? Action spectrum. What it is? It is the rate of physiological activity. What it is? It is the rate of physiological activity plotted against the wavelength of light. Against what? Against the wavelength of light. Clear? So here also you can see y-axis, x-axis. Along the x-axis we have taken the different wavelength of light in the form of nanometer, 400 to 700 nanometer we've got. Against that, rate of photosynthesis. So this peak, it is showing the rate of photosynthesis. Here the physiological activity means photosynthesis. We are talking about the photosynthesis. At which wavelength photosynthesis is maximum? At which wavelength there is an action? So that spectrum is called as what? Action spectrum. So here you can see the peak. This peak is lying at which wavelength? Can you tell? Look at this wavelength. So where is this peak? It is near to 400 nanometer. At 400 nanometer, plant shows high rate of photosynthesis. Then it gradually decreases as it passes the 500 nanometer. Then gradually it increases towards the 700 nanometer. Right? Can you correlate with this graph with some other spectrum? Just now which spectrum we have studied previous to this one? We have studied about the absorption spectrum. Absorption spectrum, we can see the chlorophyll A molecule is showing maximum absorption towards the 400 nanometer. Right? And here the rate of photosynthesis also maximum towards 400 nanometer. That means there is a correlation between the absorption and rate of photosynthesis over here. So where there is a more absorption, there is high rate of photosynthesis, obviously. Then this particular peak, it is matching with the chlorophyll A pigments graph. Clear? And once again, if you see further, this peak is not only here, but also you can see one more peak nearby the 700 nanometer. This peak can be seen here in absorption spectrum of chlorophyll A molecule, 700 nanometer. So here also peak is there of chlorophyll A. So maximum absorption is there. Hence the rate of photosynthesis is also maximum towards the 700 nanometer. So action spectrum and absorption spectrum, if we superimpose this rate of photosynthesis, if we superimpose this action spectrum on this absorption spectrum, then how it looks like? Okay, let us see how it looks like. What we are going to do now, we are going to superimpose. Why we are going to superimpose? Because these peaks are showing similarity with the peaks of chlorophyll A molecule, right? So we will get a clear idea about the relationship between the absorption of light as well as the rate of photosynthesis, that is the relationship between absorption spectrum and action spectrum. Clear? So let us superimpose this action spectrum on absorption spectrum. Why? Because there is a similarity. Between these two spectrum. Okay, as I already told you, the similarity of the action spectrum of photosynthesis and the absorption spectrum of chlorophyll tell us that the chlorophylls are the most important pigments in the process. Clear? So here if you see this graph showing the action spectrum of photosynthesis, which is superimposed on absorption spectrum of chlorophyll A. So here it is a absorption spectrum of chlorophyll A molecule. Just we have taken only of chlorophyll A molecule. We have discarded the chlorophyll B and carotenoid peaks. So this peak is of absorption spectrum of chlorophyll A. You can see the peak lying towards the 400 nanometer as well as towards the 700 nanometer. At this particular nanometer, we see the rate of photosynthesis is maximum. Where the chlorophyll A molecule is having the maximum absorption, there only the rate of photosynthesis is maximum as well as towards the 700 nanometer. Right? So moving to the next point. In VIBGYOR, what is this VIBGYOR? It is a spectrum. It is a what? It's a spectrum. Violet, indigo, blue, green, yellow, orange and red. When we get this spectrum, when we pass a light through prism, it splits into different colors. So in VIBGYOR spectra, chlorophyll A shows maximum absorption. Which pigment is showing maximum absorption? Chlorophyll A, please remember it. And hence the rate of photosynthesis is the highest at the blue and the red region. So the plants, they show high rate of photosynthesis in blue and red region. Which region? Blue and red region. Clear? Apart from this, the range of the colors, the rate of absorption, rate of photosynthesis and the maximum absorption is towards blue and red region. Clear? So moving to the next one. So because of this, chlorophyll A is the main pigment in the photosynthesis. So among all the type of pigments, chlorophyll A, chlorophyll B, xanthophylls, carotenoids, the chlorophyll A is considered as the main pigment. Why main pigment? Because maximum absorption and maximum rate of photosynthesis is seen in case of chlorophyll A. So hence chlorophyll A is the main pigment in the photosynthesis. And what about the other pigments? What about the other pigments? Other pigments are considered as accessory pigments, extra pigments. So what are those pigments? Chlorophyll B, xanthophylls and carotenoids. You should remember this. Chlorophyll A is the main pigment in photosynthesis, whereas chlorophyll B, xanthophylls and carotenoids, they are considered as accessory pigments, extra pigments. You can ask me question like, sir, if it is a main pigment, then what is the role of these accessory pigments? Why they are present? Because here let us have a look at the significance of accessory pigments. So accessory pigments absorb a wider range of light. Chlorophyll A is absorbing only somewhere 400 nanometer and 700 nanometer. Whereas the other pigments, they are absorbing the different wavelength. They absorb wider range of light and transfer the energy to the chlorophyll A and transfer those energy towards the chlorophyll A. As well as it protects the chlorophyll A from the photooxidation. What happens? These pigments, they are going to absorb the light energy. So after getting the light energy, after absorbing the light energy, they get excited. They get what? They get excited. So because of this, the electron, they are going to lose the electron. Oxidation is going to take place. What type of oxidation? Photooxidation. Because of the light, light oxidation, loss of electron. So if they lose the electron, they are going to get damaged. They are going to get destroyed. So in order to protect it from the photooxidation, these accessory pigments are helping or protecting the chlorophyll A molecule. Clear? So because of that, we need accessory pigments. Not only it is protecting chlorophyll A, but also they are covering the wide range of light. Plants, they just don't want only at 400 or 700 nanometer. Apart from that, different wavelengths are also there for the different pigments. Clear? Okay, now moving to the next one. Okay, now let us study about overall process of photosynthesis, right? So overall photosynthesis process can be studied under two phases. How many phases? Two phases. What are those? Light reaction and dark reaction. Very simple. Overall photosynthesis can be studied under two phases. How many phases? Two phases. Exactly. What are those? Light reaction and dark reaction. Repeat it. Light reaction and dark reaction. Clear? Okay. So now first we will study about the light reaction. Clear? Light reaction is also known as photochemical phase. What it is called as? Photochemical phase. Which reaction? Light reaction. Why it is called as light reaction? Because this phase directly depends on light. This phase directly depends on light. So there are different phases are there. So this particular phase, light reaction, is depending directly on the light. So hence it is light reaction. Pigment absorb light energy and produce ATP. ATP, energy currency, adenosine triphosphate. Which phosphate? Triphosphate. Which triphosphate? Adenosine triphosphate, which is the energy currency. Pigment absorb light energy and produce ATP. Just now we have studied. Pigments, they absorb light. So after absorbing light, what they are going to produce? ATP. In which reaction? Light reaction. Which reaction? Light reaction. It is called as what? Also called as what? Photochemical phase. Photochemical phase. Now the question is, where does this light reaction takes place? Where does this light reaction takes place? Light reaction takes place in thylakoid membrane. Do you remember where these thylakoids are present? If you don't remember, then go to my previous video. Here you will get the link. Or else you can also continue this video. Better you go through my previous video, that is part 1 of this photosynthesis, then come back to this video. You will understand. Anyhow, let's move. So first we need to find out where this thylakoid, what are these thylakoids, where they are present. What is this? What is this? Can you tell? This is the leaf. This is what? This is a leaf. Leaf, you know, they are absorbing light. They are absorbing the light. So where this light is going, where this light should be penetrated. So in order to find out that, we have to take the cross section, transverse section. So when we take the transverse section of the leaf, we will get this kind of structure where you will see the upper layer and lower layer. Upper one, it is called as upper epidermis. Lower layer, this single layer, it is called lower epidermis. So between the upper epidermis and lower epidermis, between upper epidermis and lower epidermis, you will see some cells. Cells are they mesophyll cells. We call them as a mesophyll cells. Meso, between, fill, leaf. Between the leaf, upper epidermis and lower epidermis, there are certain cells called mesophyll cells or tissues overall group. So in these cells, can you see these dots? Can you see these dots? So these dots are what? They are chloroplast, cell organelle, specialized for absorption of light energy and convert it into photon. So from where this structure is coming? From the leaf. When we take the cross section, observe under the microscope, so you will get this kind of structure. So when we zoom, when we take out this, you know, dotted structure, it looks like this. It is a chloroplast, cell organelle. It is a cell organelle. So the light which is, you know, absorbed by the leaf, actually it is penetrating deep inside, you know, it is penetrating through the epidermis and entering into the mesophyll tissues, mesophyll cells, and then it is entering into the chloroplast. It is a specialized cell organelle found in plants and some microorganisms, as well as in the lower plants like algae. Chloroplast structure we have studied, right? So it is a double membrane bound organelle. Inside you will see the stroma and some membranous system. Membranous system we have studied in our previous video. This disc-like structure, they are called as what? Thylakoid. They are in turn made up of a membrane. They have their own membrane called thylakoid membrane. This boundary you can see. First of all, the chloroplast is made up of a membrane, double membrane. In that there are certain membranous structures are there, disc-like structures, thylakoid. So whole this, you know, the disc-like structure thylakoid, they are arranged one above the other. This whole structure we call it as a grana, and the singular term is granum. So all together we call it as a granum. So grana are connected each other by a tubular structure that is called as what? Stroma lamellae. Stroma lamellae. So the light is actually penetrating into the chloroplast from the leaf. Into the mesophyll tissue, and inside it is penetrating inside, deep inside the chloroplast. Particularly where it is penetrating, where it is absorbed in the thylakoid. So if we take out this single structure, that is thylakoid, it is like this. It is having its membrane enclosing a space that is called as lumen, right? So here the membrane of this thylakoid membrane, it is having the group of pigments. So here the pigments are actually present. Where the pigments are actually present? In the plants, in the leaves, in the thylakoid membrane of thylakoid. Where these thylakoid are present? These thylakoid are present in a chloroplast, right? Where these chloroplasts are present? They are present in a mesophyll tissue, this dotted structure. Where this mesophyll tissue is present? Inside the leaf. So the light energy is actually absorbed by the thylakoid membrane, because in order to absorb the light, the pigments which are present here in the thylakoid membrane. Clear? Okay. Alright, now we came to know that these pigments, chlorophyll A, chlorophyll B and accessory pigments, they are present where? They are present in the thylakoid membrane, and they are just not present randomly. There is a system of these pigments. Basically there are two types of system are present. How many types? Two types of photosystem. We call them as a photosystem. Two photosystem, that is photosystem 1 PS1 and photosystem 2 PS2 are present in the thylakoid membrane. Each photosystem consists of light harvesting complex. There is actually the system of these pigments. They are not just present randomly here and there. They are arranged very, perfect manner. So two photosystems are there. Each photosystem consisting of light harvesting complex, short form LHC. These are nothing but pigment molecule bonded with the proteins. Pigments bonded with the proteins, forming a complex. That complex is called as what? Light harvesting complex. And where this light harvesting complex are present? In the photosystem. In both photosystem. Photosystem 1, photosystem 2. In LHC, that is light harvesting complex, it has two components once again here. That is reaction center and antenna. How many components? Two components. What are those? Reaction center and antenna. Here, reaction center consists of chlorophyll A molecule and it absorbs light. Of course, pigment, chlorophyll A, it absorbs light. Reaction center, it is going to form the reaction center because it is a main pigment. Why it is main pigment? It absorbs maximum wavelength of light, and it also shows high rate of photosynthesis. So it is a main pigment. This reaction center is different in both photosystem. There are two photosystem, PS1 and PS2. Photosystem 1 and photosystem 2. In PS1, the chlorophyll A has maximum or A has absorption peak at 700 nanometer. Hence also designated as P700. In photosystem 1, the reaction center consisting of chlorophyll A has the absorption peak at 700 nanometer. So because of that, PS1 is also known as P700. P700 here, it represents what? Nanometer. Why 700 nanometer? Because this chlorophyll molecule is absorbing, it is having absorption peak over there. In photosystem 2, chlorophyll A has absorption peak at 680 nanometer. Hence it can be called as P680. 680 means here the nanometer. Clear? So moving to the next one, that is antennae. Consisting of accessory pigments which increase the efficiency of photosynthesis by absorbing different wavelength of light. We have studied, we know that apart from chlorophyll A, chlorophyll B, xanthophyll, carotenoids, altogether they make accessory pigments, and these accessory pigments only we are calling it as an antenna because they are covering the wide range of wavelength, thereby increasing the efficiency of photosynthesis. Here you can see the diagrammatic representation. This one is a reaction center. Means chlorophyll A. Clear? So overall diagram is of light harvesting complex, LHC, light harvesting complex. So this one is the reaction center of chlorophyll A. Chlorophyll A becomes or makes the reaction center. Along with that, you can see the surrounding to the reaction center, there are other pigments. Accessory pigments forms antenna. So they are going to absorb the photon packet of light energy. So that energy is transferred from all these accessory pigment to reaction center. So all the energy which is received by the antenna or the accessory pigment is finally transferred to the reaction center chlorophyll A. Then it goes to that energy in the form of electron goes to the electron acceptor. That will cover afterwards. Is it clear? Light harvesting complex. Okay, very good. So we'll move to the next one. Here is a flow chart that will make you understand all these systems, right? So here leaf, inside the leaf, mesophyll cells or mesophyll tissue. In the mesophyll cells, chloroplast, cell organelle A, which in turn consisting of thylakoids. Thylakoid having their own membrane, thylakoid membrane. So inside the thylakoid membrane, we see photosystem. We see, the system are there. Two types of photosystem we have seen, PS1, PS2, photosystem 1 and photosystem 2. Clear? So each photosystem is having light harvesting complex. They are collecting the light energy. Light harvesting complex. Both the system, photosystem 1 and photosystem 2. PS1, PS2, both are having what? Light harvesting complex. LHC, light harvesting complex. Clear? So, light harvesting system, just now we have studied, we have seen in the diagrammatic representation, it consisting of two components, reaction center and antenna. Here also reaction center and antenna. But the reaction center is different. In photosystem 1 and photosystem 2. Photosystem 1 and photosystem 2 consisting of different reaction center. It is because reaction center here in photosystem 1 is having absorption peak at 700 nanometers. So P700, it is designated as P700. Whereas the reaction center in case of PS2, it is having absorption peak at 680 nanometer. So this makes the difference between the PS1 and PS2. PS1 absorb at 700 nanometer maximum peak. Photosystem 2, its reaction center absorb at 680. That means it is showing the maximum peak around 680 nanometer. Whereas the antenna, they remain same, consisting of accessory pigment. Here also it consisting of accessory pigment. Clear? Okay, now coming to the main event of the light reaction. What are the events are going to take place in the light reaction that we are going to see? The event is light absorption. Obviously, in the light reaction, it should be the absorption of light. Then there is a water splitting, that is also called as photolysis. Means in presence of light, water is broken down into hydrogen and oxygen. So that process, photolysis, in presence of light, water is splitted. And there is the release of oxygen. Then the formation of ATP and NADPH. Clear? So what are the events taking place in the reaction? Light reaction, light absorption, water splitting, it is also called as photolysis, oxygen release, formation of ATP and NADPH. Clear? So we'll stop here, and in our next class we'll see everything in detail. We'll understand all this process. Hope you found this video useful. If so, hit like button, share this video. If you are not subscribed yet, please consider subscribing. I will see you in my next video. Till then, take care.